This is Mr. Lidke here. What I'm going to do is I'm going to show you how to complete this protein synthesis activity if you were not in school. All right, so what you want to do is first you want to go ahead and read these 3 paragraphs. So go ahead, push pause on the video here, read the 3 paragraphs. I'll be here waiting for you. Okay. So thanks for reading the 3 paragraphs. So what I'm going to do is I'm going to go to my next page here. So what we're looking at is we are looking at the non-coding and the coding side of DNA. So these are the bases that are in the center of the double helix on DNA. And we can see that adenine and thymine always pair together. Okay, guanine and cytosine always pair together. So in this sequence, there's information to build a certain protein. And in particular, it's in the coding side. Like this is the side that has the information for the protein. All right, so go ahead and do this on yours. But if we look at the coding side, we have this TAC sequence. That is the start code. So I think about a chromosome and genes kind of like a paragraph and sentences. So of course, a paragraph is made up of smaller segments that we call sentences. Similar to a chromosome is also made up of smaller segments that we call genes. Well, there has to be something embedded in there that says this is the end of segment 1 and the beginning of segment 2. So in paragraph format, of course, it's the period that indicates the end of sentence 1, and then the capital letter indicates it is the beginning of sentence number 2. Well, this TAC sequence in the coding side of DNA, that is the genetic equivalent to the capital letter that says this is the start of the gene. All right, then what I'm going to do is I'm going to go ahead and after the start code, I'm going to start breaking up the DNA sequence into groups of 3 nucleotides. So go ahead, do that on your sheet. Break it up into 3 nucleotides. So now what we need to do is we need to figure out what is the messenger RNA sequence that would come from the coding DNA sequence. So the DNA sequence, that is the recipe for cookies written in Chinese. But of course, you or I would not be able to look at a recipe written in Chinese and actually bake the cookies. So we would need a copy of the recipe in a different language. So that is this RNA stuff. All right, so still the idea of complementary base pairing holds true. So if there is a thymine in the coding DNA side, in the messenger RNA, there will be an adenine paired with it. Okay, if there's an adenine on the coding DNA, paired with it is going to be a uracil. Okay, the language of RNA has U's, no T's. So I kind of think about RNA like the Hawaiian language, and in reality, there are no T's in the Hawaiian language. So that is the thing that makes the language of RNA different than the language of DNA. So next we have a C in the coding DNA side. So C and G always pair together. So then paired with it is going to be a G. So next we have an A, paired with it is going to be U. U, C. All right, and you should go ahead and keep yourself going there. finish out the messenger RNA. I'm just going to stop so that way I can keep the video a little bit shorter. All right. Then the next thing is, like, what we have here is the messenger RNA has codons. Those 3 nucleotides, AUG, that sequence of 3, that is a codon. The separate transfer RNA molecule is going to have an anticodon, which is going to match and pair with the messenger RNA codon. So again, it's still the idea of complementary base pairing. So if there is a messenger RNA has an adenine, on the transfer RNA, there is going to be a uracil. Again, we're dealing with RNA. The language of RNA has U's, no T's. So messenger RNA has a U. The transfer RNA will pair in an A. C, A, G. Okay, and you should go ahead and finish that transfer RNA line. Again, I'm just keeping this short to reduce the length of the video. Okay, so now I kind of draw my transfer RNAs like little pitchforks. Okay, on one end of the pitchfork is going to be this sequence of 3 nucleotides. Okay, on the other end of the pitchfork is going to be a specific amino acid. So if you think about a protein like a brick wall, the amino acid is like the bricks, and that transfer RNA anticodon of UAC is storing the code for a specific brick to put into the wall. So then what we want to do is then we're going to go to this table on front. Okay, so I'm just going to write up here, we had the sequence of UAC. So this is going to code for a certain amino acid in this table. So what we're going to do is we're going to go down until we get the first letter, U. All right. Then what we're going to do is we're going to go right until we get underneath the second letter, A. So if we look, we have U, so the blank, A, and then, so what we know is we are going to be somewhere in this box right here, based off the first 2 letters of UA. Then that's where the third letter comes in. So I go down, okay, and the third letter is a C. So we've got U, A, and then the third letter being a C. So what that would mean then is that would code for methionine. Okay, so then I'm going to go back, and in that circle there, I'm going to write met for methionine. All right, so here we will do another example here. So then our next transfer RNA has the anticodon of AAG. So we go back to our table. We have the sequence of AAG. So we want to figure out which amino acid does that code for. So again, I'm going to go down until I get to my first letter, A. So now I know that I'm going to be somewhere in this row. Then that's where the second letter comes in. So then I'm going to go right until I get underneath the second letter. So it's an A. So based on this, we know that we are going to be somewhere in this box on the sequence of A, and then the G will tell us which of these 4 in the box is specifically being coded for. So then again, we go down until we get the third letter, G. Okay, so it's kind of like, to write this out, the sequence would be AG. Well, what that's going to code for is that is going to code for PHE, which is a specific amino acid called phenylalanine. So I'm going to go back here, and I'm going to make myself another little bubble, and then in the middle here, I'm going to do PHE. All right, so you should go ahead, push pause right now, and figure out the rest of the amino acids. All right, so then once you take it off of pause, now we are ready to do the next thing. We want to know, what chemical property does this amino acid have? Okay, so what we're going to do is we are going to this table right here. Okay, so that amino acid, that first amino acid was MET, which stands for methionine. And if we go underneath the chemical property here, we see that it is nonpolar. So what that means is nonpolar means no charge. So that's why we're going to write a 0. So we're going to go back up here, and I'm going to switch my color here, and I'm just going to put a 0 for no charge. So then I want to know what is the chemical property of the next amino acid. So we go down, we look at the next amino acid, which is PHE, which stands for phenylalanine, and we notice that this one is also nonpolar, no charge. So all we're going to do is we're just going to write what's inside the parentheses right there. So we go right here, and again, we're just going to write a 0 for no charge. So what you're going to get is you're going to get a pattern of charges across here. Okay? And it is that pattern of charges that is going to cause the protein to fold a certain way. All right, and that is incredibly important because that's going to cause the protein to be a certain shape. And shapes are very important to proteins, and we will continue to deal with shape in future lessons coming up here. So you should go ahead, finish the chemical properties, and then you should also do this example right here as a practice on your own. All right, then once you're done with that, what you should do is you should do these questions. Okay, and usually I get some questions about this one. So in the space above, redraw this protein after it has folded into its shape. Okay, so if we imagine that this sequence of beads right there is on a flexible string, okay, and we notice that in here, like we have some positive ones, we have some negative ones. Well, of course, we know that positives and negatives attract. So if this thing is flexible and the negative attracts to the positive, what do you think this protein is going to look like? So that's a little guidance there for question number 2. But then, yes, you should do these other questions all the way down, okay, including question number 7 right here. So this is an important introductory activity to how DNA stores information. And that's a little bit of a introduction there. If you have any questions, you should let me know, but that is the end of this video. Thank you.